Welcome everyone. Today we are going to talk about cell transport. Now, before we begin, let's ask ourselves, what makes up the membrane? The cell membrane, actually. If you can't answer it right now at this moment, don't worry. I'll show the answer right now. Phospholipid, proteins, and cholesterol make up the cell membrane. Now, there isn't just one cell transport, there's two types of cell transport. The first type of cell transport is passive. The second one is active. Now, you can probably already guess where this is going. So passive means, basically passive has diffusion and osmosis. It goes from high concentration to low concentration. And it requires no ATP, or as we call it, energy. Imagine it like a car. You're up in a hill. When you're up in a hill and you're going to go down the hill, you don't need to put the gas pedal, right? So you don't need any energy. It's like when you're running down the hill. You don't need any energy. But with active, you need energy. Why? Because it goes from low concentration to high concentration. Like I said, imagine you're driving up a hill this time. You obviously need to press the gas pedal. And if you're running up a hill, you obviously need energy to run up the hill. Now, let's talk about the bulk transport and osmosis. So now let's talk about bulk transport. So there's three, actually. There's a phago, which basically is a cell eating, and aka solid. Pino, cell drinking, liquid. And exocytosis is secretion. Now, let's get to osmosis, shall we? So now there's three types of osmosis. There's a hypertonic, which is when you have a high concentration on the outside, the cell shrinks. Hypotonic is when you have a lower concentration outside the cell. It makes the cell grow and burst. And isotonic is when the solution has the same concentration inside and out. So this is a picture of hypotonic. As you can see, it burst, and it grew, and it's fat and chubby just like me, except the bursting part. Hypertonic, the cell shrinks because the concentration on the outside is so high that it just the inside can't handle it, so it just shrinks. But isotonic, wow, look at that. Isn't it perfect? Isn't it pretty? I love it. So now let's talk about central dogma. Well, basically what central dogma is, it describes a two-step process, transcription and translation, by which the information in genes flows into proteins. So DNA turns into RNA, RNA then turns into proteins. I will show you a picture of how this all looks like. But before I start all this, let's make sure we are on the same page. We need to know what DNA codons are. It's A, T, C, G. Now, do we know what they're paired up with? Yes. A goes with T, T goes with A, C goes with G, and G goes with C. Now, DNA is a little bit, just a tad bit different. but to be honest, it's not that different at all. There's an A, there's a U, there's a C, and there's a G. Same thing. A pairs with U, U pairs with A, C pairs with G, and G pairs with C. The only thing that changes is the U and T. But anyways, now that that's over, this is the nucleus right here. These are 2 DNA strips, and the DNA is so huge it can't go into these nucleus pores, but what is tiny enough is the mRNA. That is really tiny, so you could go through that. This is called transcription. The only way to remember is C comes before L in the alphabet. Now, the mRNA, or the messenger mRNA, goes all the way here, and it lands right here in a small subunit. If you didn't know, ribosomes are made up of 2 units, a large subunit and a small subunit. Now, here are tRNA, which is transferred RNA. Not only do they have amino acid, but they also have anticodon. Basically, what anticodon is, this part and this part. It's the opposite of what's the original one. So let's say, for example, this one here has A, C, G. Well, the anticodon will obviously be U, G, C. Now, basically this is how DNA is turned into RNA, mRNA, tRNA, and also the RNA here turns into proteins, because the way that these tRNA enter, they enter through the A, P, and the P section, that's when they put the little, that's when they connect the 2 codons together, and the E, they go out and on their way. So I hope you learned a little bit. Thank you for joining me today. I really appreciate it, to be honest. But yeah, thank you very much, and I hope I see you on the next and last video from me. Bye.